So this is a lecture on the second chapter of cardiovascular system. This is all about heart anatomy. That's what we're going to see today. Um, so the first thing we're going to talk about is where is the heart located, the structure of the heart. So we're looking into the anatomy portion of the heart, and then we will move on to the physiology portion and how the blood moves through the system and all that stuff. So, heart basically is a muscular pump. So it's made of cardiac muscle. It main purpose of it is to pump the blood coming from the body to the lungs for oxygenation and from back from the lungs to the heart, and from the heart it goes to rest of the body for oxygenation, spreading the oxygenated blood. So technically you have two different areas in the heart, the one that pumps the blood to the lungs and the other pump that's pumping the blood to the body. So you have the right side of the heart that receives deoxygenated blood from the tissue and then pumps it to the lungs. That'll be part of your pulmonary circuit. And the left side receives the oxygenated blood from the lungs and pumps it back to the body. That'll be your systemic circuit, which we'll talk about in detail. So here basically is what they're talking about, the systemic and the um, pulmonary circuit. So where is this heart located? Heart is basically located in the thoracic cavity, more specifically the mediastinal cavity. Um, and it's surrounded by a double layered membrane called as pericardium. So technically it's in the pericardial cavity of your thoracic cavity. So if you look at the heart, it looks like a sitting duck, meaning it has the tail portion, which is the apex of the heart, towards the left side. So technically it is mid-clavicular line. So if you look at that, this is your mid-clavicular line right here. That's where the heart ends at the fifth intercostal space right here. So fifth intervertebral space right there. And then on top, you have the second intervertebral or second rib or the intervertebral space. So that's basically where it is located, and the heart is tilted mostly towards the left side. For most of the people, there are certain congenital area where the dextrocardia, it's called as, where the heart will be located on the right side. So the physician should be careful while analyzing those kind of patients because their blood vessels, everything will be on the opposite side compared to the normal orientation that most people have. Anteriorly, you have the sternum. Posteriorly to the heart, you have the vertebral column. So you can see that over here. Um, the heart basically has the base, which is on the top towards the right shoulder, and the apex, which is towards the left, pointing downwards to the hip. And the apical impulse, basically, when the heart beats, can be palpated between the fifth and sixth rib, just below the left nipple area. So this is what I was talking about, the pericardial layer. Pericardium is a double layered sac. It has two layers, parietal and visceral pericardium. So this is what you're looking at. So you can see the parietal layer of the pericardium and then the epicardium, which is the visceral layer of the pericardium. So these two layers make up the top layer. So epicardium actually forms one of the layers of the heart wall, which is the visceral pericardium. And in between the parietal and the visceral pericardium, or the epicardium, you have the pericardial cavity within which you have the fluid to prevent the friction between the two layers itself. So the parietal pericardium layer is connected to the thoracic wall, and it doesn't move with the organ. So it doesn't move with the heart when the heart beats. So it stays put. The epicardium, on the other hand, the visceral layer of the pericardium, that is adhered to the heart wall itself. So it moves with the heart. So having one layer not moving and the other layer moving will create friction. That's why you have the pericardial cavity with the pericardial fluid in between it. Right underneath the epicardium or the visceral pericardium, you have the myocardium, which is the cardiac muscle. And then beneath that, you have the endocardium, which continues out through the aorta as endothelial lining of the blood vessel. So that basically are the three layers that you're looking at the heart wall. So you have the epicardium, which is the visceral pericardium, you have the myocardium and the endocardium. All three together forms the heart wall itself. And if these layers, if inflammation of the pericardial layer is what pericarditis happens, pericarditis can happen with, say, artificial valves that have been put in, or any kind of blood-borne pathogen that can end up in the heart and cause pericarditis as well. The problem with this is if when you have inflammation of the pericardium, it does not have that lubrication that is needed. So you have the rubbing of the two layers, which is what causes the pericardial frictional rub. So every time the heartbeat, it actually hurts. Um, when this layer, this cavity has been filled with other than the fluid, say you have a spontaneous puncture of the heart wall, or you have infection that's leading to inflammation, which is leading to the fluid accumulation. Anything can cause cardiac tamponade, which excess fluid is leaking into the pericardial space, which also hinder the pumping ability of the heart. So you have to remove that excess fluid so the heart can beat properly. This again is the three layers that I was talking about: the epicardium, which is the visceral pericardial layer, and then myocardium, which is the cardiac muscle, and then the third layer is the endocardium, which is the innermost layer, endothelial lining, which is basically a continuation of endocardium into the blood vessel. It has to be really smooth, like we saw in the previous chapter, where the endothelial lining produces nitric oxide and prostacyclin and keeps the platelets in its inactive form. That is part of this as well. Now, when you look at the myocardium, it is actually located in a specific pattern or bundles, which we can call it. So it's interlaced with the connective tissue to form these bundles. And these basically are important while you're spreading the impulse throughout the cardiac muscle. So you can see how you have two bundles for two atria, but you have for the ventricles one single massive bundle, so they contract together as one. So that is very important for the function of the heart itself. Let's look at the internal structure of the heart. The heart basically has four chambers. Um, the two atria on the top and the two ventricles in the bottom. Mainly the right side deals with the deoxygenated blood. So again, if you remember from your 226, we're looking at the picture. So the right side will be this side over here, and this will be the left side. So this is right, and that'll be left. So you have four chambers. The right side and the left side are divided by your interventricular septum. The ventricles are separated by interventricular septum, and the atria are separated by interatrial septum that's not visible in this picture. So you have a clear distinction between the right and left side of the heart, because the right side deals with deoxygenated blood and the left side deals with oxygenated blood. So you need to keep them separate. And you have the top and the bottom also divided by valves. So you have the atria, the right atrium, so here's the right atrium and here's the right ventricle, separated by a valve over here called as tricuspid valve. And the left atrium and left ventricle is separated by a valve called mitral bicuspid valve right there. So these valves basically prevent the backflow from the ventricles into the atria. So that's the main purpose of it. So initially the blood enters the right atrium. So this is right atrium is the first place the blood enters into. And then the second place it goes to the right ventricle. And then from there it goes to the pulmonary trunk to the lungs, and then back from the lungs it comes through the pulmonary veins and then into your left atrium, and then your left ventricle will be the sixth one, and then from the left ventricle it goes through the aorta into your body. So this is basically how the blood flow pathway is. We'll look into it once again in detail, but for now, let's look at the anatomy portion of it. So as the blood enters the right atrium, it goes through the tricuspid valve into the left or right ventricle and through the pulmonary artery, and then back from the pulmonary vein into the left atrium to the left ventricle, and then up into your aorta. And if you look at the valves now, the tricuspid valve is called as tricuspid because it has four cusps that holds together to close the opening between the atrium and the ventricle, hence tricuspid valve. The tricuspid valve, this is the valve right here, the white one. It is attached to like a pillar of muscle with the help of these threads called as chordae tendineae, and the chordae tendineae attaches to the pillar that you see over here called as the papillary muscle right there that you see, which in turn attaches to the wall of the ventricle, in which the wall has these trabeculae, these called as the trabeculae carneae over there. Um, the bicuspid is called as bicuspid or mitral valve because they have two flaps which go through again the chordae tendineae to the papillary muscle which attaches to the ventricle wall as well. So this basically is the brief structure of your internal anatomy. Let's look into detail of what these chambers have. If you look at the atrium, you'll notice that the atria have a flap like this outside, and this flap helps in accommodating a little bit more blood than normal. So this flap is called as the auricles of the atria. Atrium is singular, atria is plural. Um, and also, if you look at the atria, you'll notice there is in between in the atrial septum, right there, you have a fossa ovalis that's visible. So here's your fossa ovalis. This fossa ovalis used to be a foramen connecting the two atria in the embryonic stage. Because the lungs are non-functional in the embryo, the fetus does not breathe inside the embryonic or the womb itself. So basically, the lungs are inactive. So there is no need for the blood to go to the lungs for oxygenation. So all the deoxygenated blood goes from the right side to the left side through this little hole over here called as foramen ovalis in the fetal stage. Right before the birth, that foramen ovalis closes together to form the fossa ovalis. There's also another structure that connects the right and left side. It is called as the ligamentum arteriosum in adults, but arteriosa, it's basically a capillary, a tiny capillary that connects the pulmonary artery or to the aorta, again bypassing the lungs. We'll look into it towards the end of the chapter. What else do you see? On the surface, you'll notice that there is a sulcus right here. This sulcus is called as the coronary sulcus, where you have the coronary artery, the main one, and your veins, the great cardiac veins found over there. And also you'll notice that there are in the posterior portion, you also have another sulcus called as the posterior interventricular sulcus right here, where you have the middle cardiac vein and your posterior interventricular artery also found in that area. So you have anteriorly and one posteriorly found over there. Um, we already talked about the receiving chamber being the atria. Basically, it receives the blood from the blood vessels. Now, in your atria, you have ridges that are formed, which is called as pectinate muscle. You should see that in this over here. So here are your pectinate muscle that are found only in your atria. And in the cross section of your ventricles, you'll see these kind of like web-like structures called as the trabeculae carneae, which I already told you about. So atria has pectinate muscle, and over here you have trabeculae carneae and the papillary muscle that holds the valves to the walls. Um, Let's look at the ventricles. Ventricles are discharging chambers or the main pumps of the artery of the heart. The right pump, which is the right ventricle, pumps to the pulmonary circuit, to the lungs, which are feeble. They're thin-walled pumps. So you can see the right ventricle has a thinner wall because it's only going to the lungs. You don't need high pressure blood to going to the lungs because it'll shatter the capillaries in the lungs. So it doesn't have that much pressure generated. That'll be in your right ventricle pumping to the lungs. The left ventricle is the one that pumps blood to your body. So that's the one that has the thickest wall among the two pumps, which is the right and left ventricle. So this is the one that generates your blood pressure. So if you're looking at your arterial blood pressure, it's 120/80. So that 120 is generated by your left ventricle in your body, which is why it is more muscular compared to the right ventricle. Let's look at the valves that are found in the blood vessels. So we already talked about the valves in between the atria and ventricles. Now let's look at the valves that protect the blood vessels from regurgitation back into the ventricles. So when the ventricles pump, the blood goes from the right ventricle to the pulmonary trunk, and from the left ventricle it goes to your aorta right there. So you have two valves that protect these two blood vessels. It's called as the pulmonary valve, and then you have the aortic valve right there. The common name for both the valves are called the semilunar valves because they're half moon shaped valves. So it kind of cups the blood. So when when the ventricles are relaxed and the pressure is low, the high pressure blood tries to regurgitate back into the ventricles, but these valves actually prevent that from happening, regurgitation from happening, by pooling the blood in the cusp and tightening the cusps together so there is no regurgitation back into the ventricles. It happens both in pulmonary valve and in your aorta. Um, let's move on to your the blood pathway that I was talking about earlier. Um, So this is the blood pathway that I was talking. So initially the blood, deoxygenated blood, comes from your body to the right atrium. So what all blood vessels brings this deoxygenated blood from your body to your right atrium? So if you look at this, these are the three blood vessels that bring in deoxygenated blood to your right atrium in your heart. Um, so this will be your superior vena cava, inferior vena cava, and coronary sinus. Now, the coronary sinus, I did not mention that earlier. Um, if you look at the posterior view of the heart right here, you'll notice that this blue structure right here is your coronary sinus. This is a thin-walled vein that collects blood from your heart muscles and dumps it to the right atrium. So this coronary sinus, bringing in deoxygenated blood from the heart, dumps directly into your right atrium. So that's what you're seeing over here. So you have three blood vessels dumping deoxygenated blood to your right atrium. Two of them brings blood from your body, and one of them brings blood from your heart muscles themselves. So the superior vena cava brings in blood from your shoulder up, so the head, neck region, and shoulder area. That'll be your superior vena cava. And then the inferior vena cava, the rest of the body, deoxygenated blood from the rest of the body. So both the vena cava brings in blood from the body and coronary sinus brings in blood from the heart itself, and they dump it in the right atrium. The blood accumulates in the right atrium. As the pressure increases, the tricuspid valve opens. That's what you're seeing over here. The tricuspid valve opens as the pressure increases and fills the right ventricle. Right ventricle now starts pumping, pushing the blood up into the pulmonary trunk, which then divides into two pulmonary arteries. Now, why do I say arteries? It's carrying deoxygenated blood, but I'm calling it arteries because arteries are blood vessels per definition, arteries are blood vessels that take the blood away from the heart, and veins are blood vessels that brings in blood towards the heart. In this case, there's an exception. In spite of carrying deoxygenated blood, the blood is going away from the heart, which is why we call it as pulmonary artery. This is the pulmonary circuit, and the fetal circuit that are there in the body are the only exception where the venous blood is carried by arteries. Rest of the arteries usually carry oxygenated blood. Okay, so over here you're seeing deoxygenated blood carried by an artery towards the lungs. So, and the semilunar valve closes at once. So over here you have the lung oxygenating the blood, and then the oxygenated blood is brought back into the left atrium by pulmonary veins. Again, veins are the blood vessels that brings in blood towards the heart. Over here, an exception, it is oxygenated blood that is brought into the heart, into the left atrium more specific. Once the pressure in the left atrium increases, the valves, the mitral valve or the bicuspid valve open up, filling the left ventricle. And then the left ventricle pumps into the aorta, which then goes to the body. So this is basically the pathway of your blood movement. So you can easily say that if you want to divide the pulmonary circuit per se, you can easily divide the right ventricle, the pulmonary trunk, the pulmonary veins, all right here. Okay, one second. So the right ventricle, right here, you can divide this portion. This is all pulmonary circuit. This is all systemic circuit that you have, because right ventricle is the pump of the pulmonary circuit. A pulmonary trunk or pulmonary artery carries it to the lungs, and then you have the lungs, and the left atrium brings in blood from the lungs. So those are all considered as your pulmonary circuit. And this side, when you look at it, right atrium brings in blood from the body, and then over here the left ventricle it pumps blood to the body with the help of the aorta. So all of that will be considered as your systemic circuit. So technically in the picture, you can also include this portion right there. There's something wrong with it. So I hope this pathway was clear to you. So let me go back and explain once again. You have 3 blood vessels bringing in deoxygenated blood to the right atrium. Those are your superior vena cava, inferior vena cava, bringing in deoxygenated blood from the body, and coronary sinus from the heart walls themselves. Right atrium, then through the tricuspid, fills the right ventricle. Right ventricle pumps it through the pulmonary artery to the lungs. Lungs oxygenates the blood, sends it back to the heart through pulmonary veins to your left atrium, and from left atrium through the bicuspid or mitral valve goes to the left ventricle. From the left ventricle goes to your aorta, to your body once again. So this basically is the pathway of your blood through the heart themselves. Now, the coronary circulation deals with blood, oxygenated blood given to the heart. Now, when the heart muscles pump or contract, the body receives oxygenated blood. So when the ventricles contract, the blood is pushed through the aorta, and the body receives the oxygenated blood, and the tissues are oxygenated. Whereas when you consider cardiac muscle, they can only receive oxygenated blood when the heart is in relaxed mode, because when the heart is in a contracted mode, the blood vessels are squeezed, so the muscles do not receive oxygenated blood. So when the ventricles are in relaxed mode, the heart muscles receive the oxygenated blood. When the heart is in a contracted state, the body receives oxygenated blood. So now how does the heart walls receive oxygenated blood? They have a specialized capillaries that sends the blood, oxygenated blood to the heart walls. These capillaries are called as coronary arteries. There are two main arteries that come up from the base of the aorta. One goes to the right side, it's called as right coronary artery. One goes to the left side, left coronary artery. The right coronary artery gives in multiple small branches. The main ones are your right marginal artery, and then you have in the back where they merge with the left coronary artery, the posterior ventricular artery, which we'll talk eventually. The left coronary artery basically are um, two branches. You have the circumflex artery and the anterior interventricular artery that goes through the coronary sulcus of the surface of the heart itself. So these are the major branches that supply. The circumflex artery goes all the way to the back of the heart, and the anterior interventricular artery is a major artery that supplies to the left ventricle. So if this artery is blocked, then you are going to have a massive heart attack. So you definitely want to do bypass if this is blocked. So right coronary artery, this is basically what I was talking about, the posterior interventricular artery. Um, the right side and the left side merge together right here. That forms the anastomosis. Now, anastomosis are junctions between two blood vessels. Anastomosis can be between two arteries, between two veins, between artery and veins. Over here, you're looking at two arteries forming the junction. Why do you form these junctions? The reason why you have these anastomosis is so that you have constant supply of oxygenated blood to the heart walls. You cannot, from the time that the heart starts to form in the second month of the fetal formation to the time a person dies, the heart continuously beats. So you cannot afford the heart to stop. So that means it has to have continuous supply of oxygen to the walls. So for that to happen, you need continuous supply of blood. So even in spite of any blockage in one of the arteries through these anastomosis, you eventually get blood circulation going on. So when do you require bypass? When the major arteries themselves are blocked, where there's no form of anastomosis can help. Say, for example, you have a major block in the arterial interventricular artery. That's going to be major because there aren't that many anastomoses over there. So that is very important. These are all called as coronary arteries. Please do not mix up with cardiac veins. Those are separate ones. Those are deoxygenated blood that are carried by the veins. So coronary veins, or I mean cardiac veins, these are the veins that remove the deoxygenated blood from the heart muscles. So again, they're merging. So you start off from the small veins and then merge into larger veins and then form the coronary sinus, which then dumps it to the right atria. So from the right side, you have the cardiac veins and the small cardiac vein. From the posterior, you have the middle cardiac vein. Anterior, you have the great cardiac vein that also goes through the uh, coronary sulcus anteriorly on the surface of the heart. All of these veins merge into the coronary sinus, which then dumps into the right atrium. So those are all the blood vessels that feed the heart themselves. So you might want to go through the picture. Imbalances. What is angina pectoris? This is basically pain in the thoracic region due to the blood not reaching the heart muscles. So there's an deficiency of blood delivery to the myocardium, and the cells are weakened but not dead. That's angina pectoris. If the muscles die, then you have infarctions, myocardial infarctions or heart attack, where the cells are replaced, the cardiac muscles are replaced by non-contractile scar tissue. Let's look at the microscopic anatomy now. This we're talking about the cardiac muscles themselves, which you should be familiar from your 226. Cardiac muscles are striated, they're branched, they're basically uni or binucleated. Um, what else is there? They have a specialized organ called as intercalated discs that contains your uh, desmosomes and gap junctions. That desmosomes basically prevent the cells from ripping apart from each other when the heart muscles contract, and gap junctions allow the impulses to travel from one cell to the other without any problem, so you can have the whole heart functioning as one piece of muscle called as functional syncytium. So here's a picture of the gap junctions and desmosomes that forms the intercalated discs. And you can see over here, um, these lines that you see over here, these are your intercalated discs that are found in between the two cardiac muscles, more prominently visible in this um, slide over there. All right, so the when you look at the heart muscles compared to the skeletal muscles, skeletal muscles are voluntary. You can move and stop yourself. But in the case of your heart muscles, you actually it's autonomic, so you don't have control over it. So you don't have the triad that you see in your skeletal muscles. Again, review back to 226 if you have forgotten what a triad is. You do have T-tubule right down, but you don't have these little um, pads that used to be there in the skeletal muscles. You don't have that triad. You have a lot of mitochondria because, again, heart never stops beating. So you have a lot of mitochondria. You have what else do you see? T-tubules are very simple. There are not that many of them. You have no triads. The sarcoplasmic reticulum is very simple. Large amount of mitochondria are found in um, your cardiac muscles, one or two nuclei that are seen. So those are all the main differences that you see microscopically in them. Similarities, you still have the muscle contraction proceeding by depolarization action potential. You still have the impulses traveling down T-tubule. A release of calcium through the sarcoplasmic reticulum happens. You still have the excitation contraction coupling where the calcium binds troponin. All of these are similarities to your skeletal muscles. So for your test purpose, you might want to go through the similarities and differences between cardiac and your skeletal muscles itself. So here you're looking at the differences between these two muscles. In your cardiac muscles, you actually have two type of muscles. One that only transmit impulses. It's called as your non-contractile cells or pacemaker cells. And then you have the regular contractile cardiac muscles. We'll look into these and how they function later on in this chapter. Heart muscles contracts as unit, unlike skeletal muscles where you can have independent contraction that goes on. The calcium mostly comes from your sarcoplasmic reticulum. In your in your skeletal muscles, except in your heart muscles, on the other hand, you have influx coming from both sarcoplasmic reticulum as well as extracellular fluid calcium. So initially you have an influx of calcium coming from extracellular fluid, and then you have the release of calcium from sarcoplasmic reticulum. That's also a main difference. Tetanic contraction is not possible in the heart muscles compared to the skeletal muscles. We'll talk about it in the physiology in more detail. Heart muscles solely rely on aerobic respiration, unlike cardiac skeletal muscles where you do have anaerobic respiration for ATP source. The reason why you do not have anaerobic respiration in heart muscle is because the buildup of lactic acid can damage the heart muscles and they can basically die. So that is the problem of it. Um, So here's the table of the differences of the cardiac and skeletal muscles, which will be useful for you when you go over it. Now we're moving on to the electrical events in the heart, where you're talking about the physiology portion of the heart itself. Um, when you look at the heart, the physiology of the heart, we are talking about the two types of muscles. Um, the cardiac muscle that contracts, the contractile cardiac muscle, and the non-contractile cardiac muscle, which we call as the pacemaker cells. The pacemaker cells are autorhythmic cells. They're non-contractile autorhythmic cells that generate the heartbeats in your heart. So they basically contract, or the impulses travel through a specific pattern. We call it as a conduction, intrinsic cardiac conduction system. The intrinsic cardiac conduction system, they are non-contractile autorhythmic cells that can generate the impulse, transmit the impulse into among themselves all, and also to the contractile cardiac muscles that are found in it. So if you cut open the heart walls, you'll see these conduction system on in between the regular contractile cardiac muscles. So, how do these non-contractile cardiac or pacemaker cells generate action potential? Now, you should remember from your skeletal muscle chapter, where you saw that you had an action potential starting from the resting state, and then it goes up and then goes down like that, and you have the threshold and you had the peak, these kind of things, right? You might have remember from it. That is the skeletal muscle action potential graph. What you're looking at over here is a graph of action potential made by non-contractile pacemaker cells of your heart. These are the cells that generate the heartbeats. Now, if you look at it, they do not go into resting state at all. They briefly reach there, but then once again, they go back to what we call as pacemaker potential. Now, there are, instead of resting phase, depolarization, repolarization, and back to resting phase that you saw in the skeletal muscle. You have pacemaker potential, you have depolarization, and repolarization over here for your pacemaker cells. Now, what causes these peaks in the action potential? Let's look at the pacemaker potential and what causes this one. So, resting phase is minus 60 MV that you have in your pacemaker cells. From there, it at once, as soon as it reaches there, at once it goes to the pacemaker potential, which is due to opening of sodium channels and closing of your potassium channels. So you have opening of sodium channels and closing of potassium channels that slowly pushes it to the threshold. So this dotted line that you see is the threshold, which is minus 40. Once the threshold is reached, and then starts the peak of depolarization, which is due to the calcium influx, opening of calcium channels that causes the calcium influx and depolarization. And then once it reaches the positive over here, it goes to the repolarization phase, where the calcium channel closes and the potassium channels open, and leading it back down to its resting state briefly, only to start the pacemaker potential once again. So you might want to know all of these three phases of your action potential of a typical cardiac pacemaker cells that happen in your heart. So the calcium is coming from outside initially, and then sarcoplasmic reticulum, and that floods in the cell and then causes the depolarization to happen. And once it reaches the positive, repolarization trigger in by closing the calcium channels and opening the potassium channels, bringing it back to its resting state briefly, only to start the pacemaker potential once again. Now, this type of rhythm is generated by your pacemaker cells in your heart. Okay. Now, when you look at the intrinsic conduction system that happens in your heart, you'll notice that there are specific areas that are found in these conduction system. The main area that generates that 70 beats per minute heart rate that we normally have is due to the SA node or sinoatrial node. From there, the impulse generates down to all of the right atrium, crosses the interatrial septum and goes to the left atrium. And then right where you have the interatrial and interventricular septum, right there you have a node called as AV node, which receives the impulse from the SA node. So there is a minor delay or pause of zero point 0.1 seconds between the SA node and the AV node that receiving the impulse from the SA node. So that delay basically is enough for the atrium to contract and the ventricle to fill up the blood. So let's talk about this right now. So the impulse from the SA node goes to the AV node, and then it goes to the bundle that is found in the higher portion of the interventricular septum called as AV bundle, which then separates into two bundles or right and left bundle branches, goes all the way to the apex of the heart and goes back up and branches profusely, called as subendocardial conducting network or Purkinje fibers that innervates pretty much all of your ventricle walls, the right and the left. So this is the subendocardial conducting network or Purkinje fiber that you have. So this is the pathway of the impulse. So it starts from the SA node, goes to the AV node, bundle, AV bundle, bundle branches, and then the Purkinje fiber. There is a delay between the SA node and AV node, the 0.1 seconds, and each of these areas, the AV bundle, the bundle branches, and the Purkinje fiber, they're capable of producing their own beats, but the beats that they produce are much slower compared to SA node, which is basically by the time they get ready to generate their own beat, there's another beat coming from the SA node, so they never actually get a chance to um, to generate their own beats in a normal functioning heart. Sometimes what happens is the SA node does not function, the AV node takes over, and you have 60 beats per minute of the heart. We'll look into it later on, which is what causes arrhythmias, right? So irregular heart rhythms are what we call as arrhythmia. Atrium and ventricle are uncoordinated contractions. That is what happens when you have arrhythmias. Now, fibrillation is a condition where the heart impulse it's very random, irregular. So there is no pumping of the blood to the blood vessels. Instead, the heart actually quivers, and it never pushes the blood to the pulmonary artery or the aorta itself. That's fibrillation. In that either case, you need to have like rhythm corrected. Otherwise, you're going to have problem with pumping of the function. Um, SA node, if defective, can cause ectopic focus. This is what I was talking about, where the AV node takes over, generating 40 to 60 beats per minute. This is what we call as a junctional rhythm. Extrasystole is a premature contraction that usually results in excess caffeine or nicotine, where the heart basically, before it has enough time to relax or pump, another impulse is received from the ectopic focus, another region from the heart other than the SA node. Heart block is where there's a blockage between the SA node and AV node. AV node is defective, so it basically doesn't receive the impulse from the SA node. So in that case, the bundle branches or the Purkinje fiber kicks in, and they basically pump only like 20 or 15 beats per minute, which is not adequate for life. So, which is when you have to do an artificial pacemaker. When it comes to beating of the heart and how it is controlled, it's basically modified by ANS. Now, in saying modified, it does not control the SA node directly. SA node works by itself without any ANS influence. Even if you do not have any sympathetic division acting on it, SA node will function by itself, but it only modifies it when it needs it. So when you're working out, you want extra function of the heart. So that's when sympathetic division acts on the um, AV node and your heart muscles and SA node and increases the heart rate and pumping of the blood, everything. So you have two centers that are there, cardioaxillatory center and cardioinhibitory center. Cardioaxillatory center is controlled by your sympathetic division. Cardioinhibitory center through your vagus nerve, that's your parasympathetic division. So the axillatory center goes to your SA node, AV node, and the muscles and the coronary arteries to increase the oxygenation of the muscles. The inhibitory center just goes to your SA node and AV node to reduce the heartbeats. Um, Action potential of the contractile muscle, it's a bit different than the non-contractile muscle in the sense that you see the peak are different over here. You do not see the pacemaker potential that you saw in the non-contractile pacemaker action potential. Instead, what you have is you have a nice flat resting membrane potential, and then you have a super high peak, which is the depolarization, which is caused by fast voltage gated channel opening once the heart muscles reach the threshold. And then it forms a plateau. This plateau, on the other hand, is due to the slow calcium channel opening. And this actually is needed. This plateau phase is important to prevent a tetanic contraction in the heart muscles and prevent and make sure that the heart has enough time to fill in the blood before it contracts. So it gives that excess time so that ventricles can fill in and the atrium can fill in before it contracts. So that plateau phase is very important, and it's caused by slow calcium channels opening. And then followed by that is the repolarization. Once again, slow calcium channel closes and the potassium channels open, kicking out the potassium from it. So this is the main differences between your non-contractile and contractile one. Um, Once again, let's look at the steps of your action potential in contractile cardiac muscle. The first one is the fast voltage gated sodium channels opening, causing a spike in the voltage inside the cardiac muscles, contractile cardiac muscle. So it jumps to plus 30 at once. And then followed by that is the opening of your slow calcium channel, which causes the plateau phase. This is the reason why you have 200 milliseconds duration for the contraction, and this is the reason why you do not have tetanic contraction in the heart rate, and this is also the reason why you have enough time for the ventricles to fill in with the blood before they contract completely. And then slow channels open, and then of course you have the resting state where the calcium channel closes and potassium channels opening, kicking out the potassium. That is your repolarization phase. Skeletal muscles only action potential lasts for 1 to 2 seconds, but in the cardiac muscle, it lasts for 15 to 100 milliseconds that you have, or here, 200 milliseconds that you have. The contraction of skeletal muscles lasts for 15 to 100 milliseconds, whereas for your cardiac muscle, it's 200 milliseconds. This ensures that the efficient ejection of blood is possible and the tetanic contraction is not possible between each contraction. That's very important. Now let's look into what is EKG or ECG. EKG, electrocardiography, is to monitor the electrical impulses traveling throughout the heart. What we looked at was action potential of a single muscle contraction earlier of non-contractile muscle pacemaker cells and contractile cardiac muscles. What we're looking at when we're doing EKG is the impulses that moves throughout the heart, not single muscle, but throughout the heart. You place electrodes in different parts of your thoracic cavity, your arm and limbs, and you basically monitor which angle does the impulse travels through by recording these impulses as EKG. So that is the main thing. You have 12 lead EKG placed in different positions of your thoracic cavity and your limbs, like you see over here. You also have like 4 leads where you place them in your 4 limbs and you're able to get an decent reading. When you look into an EKG, you'll notice there are peaks, flat areas, and valleys. These are basically the peaks are your peak QRS complex, and the T peak, we call it as waves. So you have a P wave, T wave, and QRS complex that you see in your EKG. Now, EKG can determine whether the impulse is traveling through the walls of the heart normally or not. If there are any kind of scar tissue, it'll find out that. If there is any ectopic focus, it'll look into that. If there are any junctional rhythm, it will be able to see that. If there are any block between the SA node and AV node, you'll be able to see that. You'll be able to monitor if you have a heart attack or you had a prior heart attack where the scar tissue has been formed. So you'll be able to see all of those when you are looking into an EKG. Now, waves are your P wave, T wave, complex is QRS complex. The intervals are your PR interval and ST segments, and the QT intervals that you have in your electrocardiogram. What is happening in each wave is what you should know. When you're recording a P wave, what actually is happening in your heart? When you notice that over here, what you're seeing is when the P wave is happening, the impulse from the SA node, it's slowly starting to spread between the atria, crosses the interventricular septum, and goes to the left atria over there. That's what you're seeing at the end of the P wave in your PQ segment that you see over there. At the end of the Q, you'll see all of the atria has been depolarized. And then you start the QRS complex. So from the QRS complex, you'll notice that the impulse is traveling now, has from SA node, it's reached the AV node into the AV bundle and bundle branches, and it's going to the apex. From the apex, it goes to the Purkinje fiber. So the end of your QRS complex, you have the entire ventricles charged, or we call it as depolarized and ready for contraction. So that'll be your next phase. So contraction happens, and then what happens? Relaxation should happen. So now you're having repolarization, right? So after depolarization, you have repolarization happening. So at the end of QRS, you have the complete depolarization of the ventricles. And at the P, or it should be T wave, not P. At the end of T wave, you have your beginning of a repolarization at the apex of the heart, and then it kind of consumes the entire ventricle when the end of the T wave that you see. So this labeling has been shifted. The P wave should be here, QRS should be here, and the T wave should be where the P is. So just FYI. Okay. It might be correct in your PowerPoints. So this is exactly what is happening in each of the readings when you look into your ECG readings. So you need to know what is happening in P wave. P wave is atrial depolarization. QRS complex is ventricular depolarization. T wave is ventricular repolarization. Atrial repolarization is not visible in your ECG because it's being masked by your QRS complex, so you don't see that in your ECG. So this is a standard ECG 12 lead standard ECG lead where you see in different angle of your heart, different positions. So different degrees of your heart. So 90 degree, 120 degree, those kind of degrees of your heart. You're monitoring the electrical impulse traveling in all these degrees and different angles of your heart. And that's what your 12 lead ECG is showing normally. Okay. Again, once again, it can detect a number of problems in the heart and heart conduction system. The thickening of the left ventricle can be also seen over there. Prolonged QT interval basically is wrist ventricular arrhythmias. Enlarged R waves is due to the enlarged ventricle, hypertrophy of the ventricle, like you see in high blood pressure patients. Elevated or depressed ST segments is your cardiac ischemia that is happening. So here what you're looking at is enlarged R waves, and then which is basically having a thicker wall of the left ventricle. Here's a picture of elevated and depressed ST segments that you see when you have a myocardial infarction. This is a normal sinus rhythm that you see. You see P wave, QRS, and T wave over here. This is a junctional rhythm where the P wave is missing. So that means the impulse that is coming to the ventricles, the impulse that's coming to the ventricles is coming from an AV node, not from an SA node, which is why the P wave is missing in this. That's very important. So AV node only beats 40 to 60. That's what you see in junctional rhythm. Here is a second degree heart block where every other beat from the SA node is transmitted through the AV node. So you'll notice P wave is here, P wave is here, but the QRST complex is missing. P wave is here, and then P wave is here. Again, QRST complex is missing. So every other one, you're missing the QRS wave, which means the AV node is not conducting every beat that has been sent by the sinus SA node, but it's only sending every other beat to the ventricles. So that'll be your ventricles are being stressed out and they're not pumping enough blood to the body. Here's a picture of ventricular fibrillation where there is no proper beat at all. The heart walls are just quivering. There's no pump per se in the heart. They're just quivering, so the blood remains stagnant. There's a high chance of clot forming in the heart when you have ventricular fibrillation. So those are the imbalances of EKG. Now let's look at the entire mechanics of the heart, or from the time what happens and the blood fills in, which valves closes, what type of ECG is recorded, what are the sounds that you hear, and then basically the pressure building in the chambers. We're going to look all of it in the mechanical events of the heart. So this is all table form, and I'm going to go into this portion that you're seeing here. When you look at the mechanical events of the cardiac cycle per se, you'll notice that the uh, you'll notice that basically there are different steps to the cardiac cycle. The first step is the ventricular filling phase, and the second one is isovolumetric contraction phase, and the third one is a ventricular ejection phase, and then fourth is isovolumetric relaxation phase. So what happens in each of these phases, and what happens with the blood, the pressure, the valves opening, ECG, and all of those. That's basically what you're seeing in the cardiac cycle. Let's look at the first phase called as ventricular filling phase, which is subdivided into two phases, passive, and we call it, and the second one is atrial contraction. So during the ventricular filling phase, the three blood vessels, the vena cava superior and inferior, and your coronary sinus, all three of them bring in deoxygenated blood to the right atrium, and the pulmonary vein brings in blood to the left atrium. So the blood is filled the atrial chamber. Now, as the blood is increasing in the atrial chamber due to pressure, mere pressure, 80% of the blood, mere pressure, the valves, AV valves, atrioventricular valves, that is your bicuspid and your tricuspid, opens, and mere due to pressure difference between the atrium and ventricle, 80% of the blood fills the ventricles initially. That is your passive phase. The rest 20% of the blood from the atrium is contracted through the atrial contraction, is pushed from the atrium into the ventricle. So atrial contraction is happening over here at the atrial contraction phase of your ventricular filling phase. So when does the atrial contraction happen? Atrial contraction happen when you have atrial depolarization happening. So ventricular filling phase, you'll notice that there is a P wave. Because the blood is moving from the atrium to the ventricles, the pressure from the atrium reduces slowly, which is what you see, the pressure difference. The right atrium, left atrium, the pressure basically is decreased. The volume in the ventricle slowly increases as the atrium starts filling the blood into the ventricles at the end of ventricular filling phase. Now, the second phase is isovolumetric contraction phase. All of the valves are closed briefly when the blood, after the blood fills the ventricles. So you have your tricuspid, bicuspid closed, you have the semilunar, pulmonary, and aortic valves closed. The blood is in the ventricles, and the blood is starting to build up the pressure, which causes the pressure inside your ventricles to rise. So that's what you're seeing over here. The left ventricle is beginning to rise. So the pressure is building. The muscles have been depolarized. So what you're seeing over here is the QRS complex, or ventricular depolarization, is being noticed over there. And then followed by the isovolumetric contraction phase is your ventricular ejection phase. The pressure starts to build up and then they start to contract. So the end of QRS complex basically is where you see your ventricular ejection phase, causing the muscles of the ventricles to contract and pushing the blood into either your pulmonary trunk from the right side or aorta from the left side. And that'll be your ventricular ejection phase, causing the blood to push up into the arteries. So you have your semilunar valves open. So the pulmonary valve and the aortic valves are open. Excuse me, which because of the pressure that builds up, causing the ventricular depolarization and contraction of the ventricles, eventually pushing the blood into your pulmonary artery, or pulmonary artery and the aorta, and slowly the blood moves in the arteries. And then what you see over is the next phase, the last phase, where the blood is still in the blood vessels. So you see the blood vessels in your aorta and in your pulmonary artery, but the ventricles are emptied out, and it is starting to ventricular repolarization phase is beginning over there. So you're seeing the T wave over there, and because the blood has moved into the aorta and pulmonary trunk, the pressure is lowest in your left ventricle. The pressure is highest in your aorta, that you see in the green over there. So the blood is trying to get back to the left ventricle, but the semilunar valve of your aorta is closing tightly, preventing the regurgitation. So it causes a little dip called as dicrotic notch, where the blood briefly goes into the cusps of the aortic valves and shoots back up into the aorta, causing the brief dip in the pressure in your aorta, which is called as dicrotic notch. And once again, this is your isovolumetric relaxation phase that you see when the blood is in the aorta and the ventricles are briefly relaxing. And you'll notice in the atrium the blood is starting to fill back again. So this cycle keeps on going and going and going. Okay, so here are the volumes that happens in the the ventricles are starting to fill up, so the volume increases to 120 ml, and then during the ventricular ejection phase the volume starts to go back down, and you have a little bit of blood remaining in the end of your isovolumetric relaxation phase. That basically is what we call as ESV. And the increased amount of blood, the highest level of blood, 120 ml, is called as end diastolic volume, or EDV. So the 120 is called as end diastolic volume, end of diastole, which is when the blood is filled in, and end of systole, end systolic volume, which is left behind blood in the ventricles, is usually 50 ml. So technically 120 minus 50 is 70 ml of blood is what is received by the aorta at the end of your ventricular ejection phase. So all of this is what we consider as cardiac cycle that happens in your heart. So we were talking about briefly the sounds. Before we get to the sounds, I want to talk about how long the heart spends in contracted phase and how long does the heart spend in a relaxed mode. So if you take an entire cardiac cycle, it only lasts for zero point 0.8 seconds. Out of that 0.8 second, the atrial contraction is 0.1 second, ventricular contraction is 0.4 seconds. So the contraction itself is only 0.4 seconds. 0.1 second for your atrium, 0.3 seconds for your ventricles. The rest of 0.4 seconds of your cardiac cycle, the heart spends in relaxing mode, quiescent period. It's heart relaxation mode, where it's just basically relaxing in the body. All right, let's look at the heart sounds. The lub-dub sound that you hear when you place a stethoscope against the thoracic cavity is due to the closing of the heart valves. There are 4 heart valves that you have, right? So heart itself has 2 valves, the atrioventricular valve, that'll be your bicuspid and tricuspid. Tricuspid between the right atrium and the right ventricle, bicuspid between the left atrium and left ventricle. And then the 2 semilunar valves between the ventricles and the blood vessels. So between the right ventricle and the pulmonary artery or the pulmonary trunk, you have the pulmonary valve, and between the left ventricle and the aorta, you have the aortic valve. So when these valves close, that's when you hear the sounds. The first sound, S1, lub sound, is due to the AV valves closing. That'll be your bicuspid and tricuspid closing. When those valves close, the blood is actually in the ventricles. So they are in, if you look at this cardiac cycle, they're actually in isovolumetric contraction phase. The valves, AV valves, close. That's when you hear the first sound of your heart beat. The second sound of your heartbeat, the dub sound, lub dub, S1, S2, the S2 sound is heard only when your semilunar valves close. So the aortic valve and pulmonary valve closes. That's when you hear the dub or S2 sound. When do they close? At the isovolumetric relaxation phase. That's when you hear the S2 sound. So there's a pause between lub dubs, which indicates the relaxation phase of the heart. That's the 0.4 seconds where the heart is relaxing. So again, the sounds of your heart is not due to the valves opening, but it's due to the valves closing. The first S1 sound is due, the lub sound is due to the ventricular AV valves closing, the bicuspid and tricuspid. The second dub sound, S2 sound, is heard by your semilunar valves closing. Now these sounds, you should have a clear sound heard when you place the stethoscope throughout the thoracic wall. But the valves are predominantly heard in certain parts of your thoracic cage. Depending on which valve, you have certain areas that are more predominantly heard, and this is the picture of those things. So here's your tricuspid valve right here, the yellow one. So the sound goes this direction towards the fifth intercostal space on the right sternal margin. And then you have your pulmonary trunk going upwards like that. So that means the pulmonary valve will be heard at the second intercostal space and second sternal margin. The mitral valve, it will be heard at the apex because it transmits downwards like that, because that's your mitral, the bicuspid valve that you have. So it goes to the fifth intercostal space along the midclavicular line. And then the aorta, it goes upwards like that. So it's going to be heard on the second intercostal space on the right sternal margin over there. So it's kind of like a crisscross area based on where these valves are located. So in these areas, you'll hear the valve sounds more predominantly than other areas of that. You will hear them, but you'll hear it more predominantly, especially when these valves are faulty. There are two kinds of faults that can happen in these valves. One, they're stenotic, where they do not open and they're calcified. And two, where they're flabby, that they just back and forth flabby. So it allows the blood go back into the atrium or go back into the ventricle, depending which valves you are talking about. So when you have these faulty valves, you hear abnormal heart sounds. We call it as heart murmurs. They're either flabby, we call it as incompetent or insufficient valves, allowing the backflow of blood and causing the whooshing sound when the chamber is relaxing. So say, for example, from the atrium, you have blood going into the ventricles. The tricuspid valve, for example, closes, should close and prevent the backflow into the atrium. But if the tricuspid is flabby, insufficient, incompetent, then when the right ventricle contracts, instead of going to the pulmonary trunk, it goes back into the right atrium. You have a whooshing sound of regurgitation when the ventricles contract. So that is basically when your AV valve are basically not working properly. The other one is stenotic valve, where they're calcified. They don't open up enough for the blood to pass through them. So you're going to have a high-pitched clicking sound heard when you have a stenotic valve in your heart. So all of these can be heard depending which valve is faulty. You can hear those whooshing sound or clicking sound more predominantly in one of these areas depending which valve is affected. So that concludes the cardiac cycle portion of it. We are now going into the volume and how much blood is pumped through the heart, and all of those. Regulation of pumping. So when you take a heartbeat, okay, one heartbeat is basically heart rate times the stroke volume per minute is cardiac output. So cardiac output is amount of blood that is pumped by each ventricle in a minute. So how much blood is pumped by your ventricles per minute determines your cardiac output. So what determines the amount of blood pumped by your ventricle in 1 minute? Heart rate, how many times the heart beats, and stroke volume, how much blood is pumped out of the heart. What is stroke volume? Stroke volume is the blood pumped out by ventricle each beat is stroke volume. Now, when we were talking about the cardiac cycle, right here, I mentioned EDV and ESV volume. EDV was 120 ml end diastolic volume. ESV was 50 ml. So I said EDV minus ESV, 120 minus 50, which is 70 ml, is what actually reaches the aorta. So if you go back once again over here to stroke volume, you'll notice that stroke volume is how much blood is pumped into the body per beat, by the ventricle per beat. So that means 120 minus 50, 70 ml of blood is pumped into the aorta per single beat. That will be your stroke volume. So 70 ml is your stroke volume. How many times your heart beat depends on your SA node. That'll be your 75 beats per minute. So heart rate times SV, 75 times 70 is converted in from milliliter to liter is 5.25 liter per minute will be your cardiac output per during rest. The cardiac output will increase depending whether you're working out or resting. If you are an athlete, the cardiac output can increase to 35 liter per minute. If you're a non-athlete, it goes up to 20 to 25 liter per minute. So the excess is basically what we call as cardiac reserve. Okay, so 5.25, it jumps to 25 liter or 35 liter. That excess that you have is cardiac reserve. Difference between resting and maximal cardiac output. What does it depend on? The cardiac output depends on stroke volume and your heart rate, directly proportional to your heart rate and your stroke volume. So whatever changes the heart rate and stroke volume will ultimately change your cardiac output. So what are the regulation of your stroke volume? Stroke volume depends on three main factors: preload, contractility, and afterload. Okay, we all know SV is equal to EDV, that is 120, and ESV, which is 50, left behind blood. Excuse me. So that'll be 70 ml per milliliter. So it depends on three things: preload, contractility, and afterload. What is preload? Preload is degree of cardiac muscle that are stretched just before they contract. So the relationship between preload and stroke volume is called as Frank-Starling law of heart. Okay, so this is basically important for you. Cardiac muscle, you need to know the definition of that. Cardiac muscle exhibits the length-tension relationship, meaning the cardiac muscle are shorter, which means it dramatically increases the contractile force. The more it's stretched, the more it's going to have the tension. That is basically what preload is. And it also depends on what causes that stretching of the cardiac muscle. Stretching of the cardiac muscle depends on how much blood goes into these chambers. What determines how much blood goes into the chambers? Venous return. So how much blood is coming back from the body determines how much stretch is going to happen in the cardiac muscle. Okay, we're talking about the right side of the heart mainly. So venous return, how much blood comes back into your right atrium is determined by the venous return. So what increases the venous return? Working out, exercising, muscles pumping against the superficial veins pushes the blood. Veins are considered as reservoirs of blood. Okay, so anytime, any given time, you have 65% of blood stored in veins. So when you're working out, the superficial veins are pushed by the muscles, pushing the blood upwards towards the heart. So more blood is going into the heart. Venous return increases when you work out and stretches the heart muscles, which means stretches the preload. The heart muscle stretches, Frank-Starling law, tension increases, preload increases. So that means venous return increases, EDV from 120 increases, say, to 140. SV increases, so the blood pumping out of the heart increases from 70 to say 90, which means cardiac output increases automatically. That'll be your preload. Contractility. How much forcefully does the muscles contract? Okay, the contraction of a cardiac muscle or any muscle depends on your electrolytes or ions. Over here, you have your calcium. You have your contractility caused by your sympathetic division and positive inotropic agents like thyroxine, glucagon, epinephrine, calcium, digitalis, all of those that increases the contractile rate of your cardiac muscle. So when you contract hardly, the left behind blood will be less. Okay, so the left behind blood, end systolic volume blood, that 50 ml that's left behind, will be lesser than 50, which means the blood that's pumped out will be more. So the higher the contractility, the lesser the ESV, so lowers the ESV, which increases the SV. So positive inotropic agent lowers the ESV and increases the SV. Negative inotropic agents basically increases the SV, hence in decreasing the S, uh, stroke volume. The last one that your stroke volume depends on is afterload. Afterload is how much struggle does the ventricles have to undergo to push the blood into the pulmonary artery and the aorta and into the body. That is determined by your systemic blood pressure and your pulmonary blood pressure. So, what is causing the backflow into the ventricles? That determines the afterload. The aortic pressure should be only 80 mmHg. So your normal blood pressure, diastolic blood pressure, is 80 mmHg. If it increases more than 80 mmHg, that means the left ventricle is having a hard time pushing the blood against that already high pressure, leaving more blood behind in the ventricles. So your end systolic volume from 50 jumps higher to say 60 or 70, which means the blood actually leaving the ventricles is far lesser than your 70 ml, which reduces the stroke volume. It applies to your right side of your heart as well. So afterload, hypertension increases the afterload, which increases the SV, left behind blood in the ventricles from 50 higher up, and reduces the when it increases the ESV, automatically it reduces the SV. So three things affects your stroke volume. Once again, your preload, venous return, and the stretchability of the cardiac muscle, contractility of the muscle. So how strong is the contractile muscle determines how much blood is left behind. So if the muscles are strongly contracting, the left behind, the ESV decreases, increasing the SV. And finally, afterload, how much pressure does it has to overcome in order to push the blood from the ventricles into the arteries. Increasing hypertension causes increased afterload, which results in increased ESV and reduced SV. So please go over the stroke volume. Any number of these damage or changes in stroke volume, anything that changes stroke volume will directly affect your cardiac output. Now, the next important thing that affects the cardiac output is your heart rate. Heart rate is affected by a number of things. The positive heart rate, which increases the heart rate, is called as positive chronotropic agents, and the one that decreases the heart rate is called as negative chronotropic agents. So you have autonomic nervous system where sympathetic division is a positive chronotropic, increasing the heart rate, whereas your parasympathetic decreases, it's a negative chronotropic agent. Chemicals from calcium, sodium, potassium, epinephrine, norepinephrine, thyroxine, any number of chemicals can also affect the heart rate. Other factors like your age, your position, your height, everything affects it. So here is your epinephrine, norepinephrine, the sympathetic division, increasing your heart rate by increasing the pacemaker activity, SA node, increasing the heart rate directly, and contractility also increases with epinephrine, norepinephrine. Parasympathetic, on the other hand, reduces, is a negative chronotropic agent, reducing the heart rate directly. Vagal tone, once again, is predominant under rest. So parasympathetic is predominant under rest, which is why our heart rate is 75 beats per minute in average, because parasympathetic dampens the heart rate. Otherwise, if you cut the vagus nerve, the normal heart rate will be 100 beats per minute. Bainbridge reflex is a sympathetic reflex initiated by increased venous return. So when you have high venous return, automatically that stretching of your atrium causes the trigger of your sympathetic reflex to cause the SA node to generate more impulse and cause the heart rate to increase, as well as your other changes like increasing contractility, so on and so forth. So that is atrial Bainbridge reflex. You might want to go over that as well. Hormones, like I said, thyroxine, epinephrine, all of those, norepinephrine, those things are positive chronotropic agent. Ions, calcium, potassium, imbalances can be very, very dangerous. Like, for example, hypocalcemia depresses heart rate. Hypercalcemia increases heart rate and contractility. Hyperkalemia leads to heart block and cardiac arrest. Hypokalemia is arrhythmia. Hyperkalemia is basically what they give to death row patients, or not patients, death row inmates, where they give this for cardiac arrest. It actually burns. That's why they give a numbing agent before they give this potassium chloride for cardiac arrest. Other factors, age, like I said, gender, exercise, body temperature, everything affects your influencing the heart rate. Increase in heart rate over 100 beats per minute is called as tachycardia. Decrease in heart rate below 60 beats per minute is bradycardia, which is basically desired for endurance training, but is inadequate in non-athletes for cells to get oxygenated properly. Imbalances, congestive heart failure, where cardiac output is so low that you don't have enough oxygenation in the tissue, so you end up with all kind of problems like edema and tissue damage and hypoxic condition. All of those are built up. Congestive heart failure um, can be due to persistent high blood pressure, where the myocardium basically has hypertrophy and then eventually weakens, or heart attack, multiple heart attack, or dilated cardiomyopathy. A number of reasons can cause that. It can lead to pulmonary congestion, where you're starting to build edema in the lungs, or it can lead to peripheral congestion, where edema is in the body and the organs itself. It doesn't matter which side of the heart is failing. Ultimately, it catches up because it's a closed circuit, and the blood basically goes through from one area to the other area. So the fluid buildup also catches up with the other area eventually. The last portion of the chapter deals with the developmental aspect of it. Heart muscle, being a muscle, it's basically from mesoderm, just like connective tissues are. Pumping starts in day 22. Okay, so the heart actually starts actively pumping in day 22, but the chamber keeps developing eventually after day 22. So initially you have two tubes that are there formed. So here are the two tubes that are formed that merges together and bulges, and this is when the heart starts pumping over here in day 22. They start bulging up, and then eventually they start twisting and bending and elongating and to form the actual heart itself. And because of this formation that you end up forming, it leads to a lot of issues in leading to congenital defects in the heart. You also want to talk about the tube bypass mechanism, where the right side deoxygenated blood bypasses the right side and lungs and goes into your left side, because the lungs are non-functional. You have the foramen ovale and you have the ductus arteriosum. Those are the two things that the deoxygenated blood bypasses from the right side and enters bypasses your lungs and then enters your left side, so the placenta can oxygenate the blood through the mother directly. So foraminovalle bypasses the lungs by taking the blood from the right atrium to the left atrium. Ductus arteriosus takes the blood, whatever left behind blood from the right ventricle enters the pulmonary trunk. It goes through the ductus arteriosus to your aorta. So two of these structures completely removes the blood from the lungs. So lungs receive only, in the fetal stage, I mean, lungs receive only oxygenated blood from the aorta. So otherwise, everything is bypassed from the lungs because they're inactive in the fetal stage. Right before the birth is basically, shortly before the birth is when these two, foraminovalle and ductus arteriosum, is converted into fossa ovalis and ligamentum arteriosum. If they remain open, then you're going to have problem in the newborn. You're going to have that blue baby syndrome where when they exert themselves, when they're crying and exert themselves, they turn blue because there's a mixing of deoxygenated blood and oxygenated blood because lungs are functioning now. There is no placenta, so there should not be any mixing of the right side and the left side blood, deoxygenated and oxygenated blood. Okay, so let's look into the congenital defects that can possibly happen. The first one is the ventricular septal defect. It's obvious there's a hole in the septum because the pressure from the left side is higher compared to the right side of the ventricle. When the ventricles contract, the more blood from the left side goes into the right side, and some blood from the right side goes into the left side. So you have mixing of deoxygenated and oxygenated blood. So you're going to see again the blue baby syndrome. Usually they close up after some time. But if they don't close up, then you have to have a surgery. It happens 1 in every 500 births, which is relatively common. And then next one is coarctation of aorta. So the coarctation of aorta, you'll notice right over here, there's a narrowing of the aorta. Now this is going to increase the pressure after load. When we are talking about the stroke volume and the factors that affect the stroke volume, we said afterload, increase in hypertension, right? Hypertension. This is artificially increasing that pressure. So you're going to end up when the left ventricle pushes the blood into the aorta, it's going to come across this narrowing of the aorta, which is going to cause the overworkload of the left ventricle, causing the hypertrophy of the left ventricle. So that has to be also surgically corrected. That is coarctation of aorta. The last one we're going to talk about is tetralogy of Fallot, where you have 4 defects in one heart. So you're going to have a ventricular septal defect, which is so high. You're going to see this defect is so high close to the aorta. Okay, so you have a ventricular septal defect, you have hypertrophy of your right ventricle, you have your coarctation of your pulmonary narrowing of the pulmonary trunk, and aorta opens from both right and left ventricle. Now, what does that mean? It means that the ventricular septal defect is so high that it's close to the opening of the aorta that aorta receives blood from both right and left ventricles because of the ventricular septal defect being so high. So that means you have severe mixing of deoxygenated and oxygenated blood. And then the next thing, why do you have hypertrophy of right ventricle? Because of the narrowing of pulmonary trunk. And also you're receiving blood from the left ventricle as well. So you have too much workload going on. So all of these condition is what tetralogy of Fallot is. 4, tetra is 4, Fallot is the guy who found it. So 1 in 2,000 births can have that, again, surgically has to be repaired when it happens over there. Um, the final thing is age-related issues. Mainly, it's because of fat deposition, atherosclerotic plaques in the coronary artery, which leads to heart attack, which leads to non-contractile connective tissues being formed instead of cardiac muscles. So that is the main age-related issues you will have. Regular exercise, eating healthy, can and stress obviously reducing stress level can actually keep your heart hale and healthy. Fibrosis of heart muscles can lead to stiffened heart, arrhythmias can happen, valves can not function properly because of stenosis. Those can all happen as you get older. It is obviously heart pump, so as you get older it kind of wears off. This basically concludes your chapter 18, um, which is the heart lecture.